I'm Nancy Horton, and I'm from the University of Arizona, and I wanted to give a few minutes to introduce the motivation for this session. Okay, so the basic structure of this session, I'm going to give a 5-minute introduction and go into my talk, and then we'll have a series of talks after me. So the subject of this session is filament formation by enzymes, which is kind of an emerging idea field for regulation of enzyme activity, but it's actually been known for quite a long time that enzymes, metabolic enzymes, can form filaments. So shown here are electron micrographs taken in the 60s, 70s, and 1980 of enzymes purified from natural sources, and you can see that they do in fact form filaments. But the effect on enzyme activity wasn't known, and it wasn't even known if these were biologically relevant or maybe just an in vitro artifact. So why did we seem to forget largely that these enzymes can form filaments? Well, I blame X-ray crystallography. Nothing personal, but I am an X-ray crystallographer. And it's a great method to see high resolution structures of proteins and enzymes. But you only see what can crystallize, and filaments are not very good at crystallizing. These are the very same enzymes that I just showed you form filaments, yet in the crystal structure, we don't see filaments. They are oligomeric. That's what the different colored chains is showing you, and that's pretty common in filament forming enzymes. But you don't see the filaments in the crystal structure, so it was almost as if it was forgotten. There were a few labs that continued to study it in their own enzyme system, but interest really exploded in 2009 and after due to these large-scale screens of protein and enzyme localization in cells. So these are using GFP or some other fluorescent protein or marker to visualize the localization of proteins in a cell, and systematically, one by one, each of the proteins in a proteome was investigated this way using fluorescence microscopy and things like yeast and Drosophila and bacteria. And surprisingly, they observed these reversible self-assemblies, often also called filaments because of their structure. They were observed in cells, so they aren't amyloid because they're reversible and they form under particular conditions. In a recent review, I tried to assemble the names and references for all the enzymes I could find that form these reversible self-assemblies in cells. And I list them here, and you can see that it's very widespread in different metabolic and biological pathways. It's conserved evolutionarily. It's found in all branches of life. But what is the relationship between the in vitro nanometer scale filaments that we see in electron microscopy and the self-assemblies of enzymes seen in cells? That's still a really interesting question. It does appear that many of the enzymes that form filaments in vitro, and the white is the cryo-EM envelope, also form these self-assemblies in cells. The green is the X-ray crystal structure superimposed into the filament so that you can see it. The X-ray crystal structure is the building block for the filament, I should say. So very similar, or the same conditions give rise to both. So it seems possible that these self-assemblies are composed of filamentous enzymes, but the way that the filaments assemble in the self-assembly is not known. How many enzymes actually form these filaments in the nanometer scale that we can see by EM? I could find 24, again, I cover in my review. Again, all different metabolic and biological pathways, all branches of life. So a conserved phenomenon that's widespread. You can see the structures vary. There's right-handed helices, left-handed helices. They can form tubes or linear structures. But what is the purpose of this? These are some interesting questions, I think. People have put forth ideas that I review in here, and my lab has done so as well. So in modulating enzyme activity, turning it on or off is a possibility. Controlling enzyme location, or in sequestering during cell division is another idea. Protection from oxidation, buffering levels of active enzymes is another idea. So, and it may be that there are distinct purposes for each different enzyme. Another question, what's the relationship between the cellular self-assemblies and the filament is an interesting question. So I'm interested in the structural relationship as well as the functional relationship. So now I'm going to talk about my system. It's an enzyme called SgrA1, which is a type 2 restriction endonuclease from the bacterium Streptomyces griseus. It cleaves this 8 base pair sequence in duplex DNA where the line is located. The R means any purine and the Y means any pyrimidine. If there's only a single copy of this primary sequence in a plasmid DNA, one finds that the enzyme will cleave it, but really slowly. But if you have multiple copies, all of these sites will be cleaved very rapidly, 200 times more faster. Really interesting is that the enzyme will cleave these secondary sites that differ from the primary by 1 base pair, either the first or the second. But it won't cleave them unless there's also a primary site present on the same DNA. It can work in trans. You can get the enzyme to cleave these if you provide enough primary site oligo in high enough concentrations and high enough enzyme concentrations. But if it's on the same plasmid DNA, you get rapid cleavage of all sites. So it appears that primary site sequence is changing the activity and the substrate specificity of the enzyme. And through a lot of biochemical and biophysical investigations, we were able to find that the enzyme forms filaments with activated DNA cleavage properties. So we put together this mechanism based on our data. So when the enzyme is bound to primary site, it's prone to form a filament, which activates it for DNA cleavage. But when it's bound to secondary site, only 1 base pair different, it doesn't filament. If there are enzymes bound to primary site around, that can induce filamentation that drags in the enzymes bound to secondary site, inducing them to cleave DNA. Incidentally, I should have mentioned the SgrA1 binds to secondary site DNA with really high affinity, nanomolar affinity, but does not cleave it until it's in the filament form. So we determined the structure of the filament in collaboration to now 3.5 angstrom resolution. My lab determined the crystal structure of the enzyme, and it looks like other type 2 restriction endos. It's a homodimer that binds to 1 duplex of DNA. And we found that the filament is a left-handed helical filament with about 4 copies of the enzyme per turn. And we find that these can grow quite long, 30 or more copies of the enzyme. But what we want to know now, what I'm going to talk about today is, and I'll talk about the structure tomorrow actually in the session called shape-shifting. But today I wanted to talk about some other studies that address these questions. So we're trying to get at why form this structure. Is there some specific advantage to controlling enzyme activity using this filament structure? To do that, we used a series of kinetic measurements and global model fitting, computational model fitting. And we used the program Kinetic Global Kinetic Explorer to create this model and to fit our data so that we could extract each of these rate constants to the pathway. This is a very, very simplified schematic of the reaction pathway because we know the filaments can get quite large. We don't assume concerted DNA cleavage like shown here. It's just to give a simplified view of it. So because in this software we had to write explicitly each equilibrium reaction, it got quite complicated with all the combinations and permutations. So we were limited pretty much to a filament size of 5 or less. To see if that was adequate to fit our data, we also constructed models that went up to filament sizes of 4, 3, and 2, and we got pretty much the same results, and that's probably because the enzyme concentration we were using in our assays limited the filament size. So here's some of the data that we fit. It's FRET data where we followed the cleavage of the reporter DNA, the unquenching of the donor with time, with the DNA on separate molecules. This step, assembly of the filament, is rate limiting, so you can change the concentration of the enzyme bound to DNA and increase the speed of DNA cleavage and get more data on the pathway that way. We also measured just DNA cleavage using denaturing gel electrophoresis, and finally having donor and acceptor FRET pairs on different DNAs to watch the assembly of the filaments and then disassembly. So we were able to fit and get good values for all of these rate constants, except for this one, but we do have the equilibrium constant, and this is pretty fast and didn't limit our reactions anyway. So here are the rate constants that we got with in the error bar limits. So I could say a lot about these and have so in this paper, a set of 2 papers actually published in JBC. But first I wanted to tell you one thing we did. We wanted to try to validate these numbers with some separate piece of data. So we used our model, our computational model, and these rate constants, and we tried to recapitulate the distribution of filament sizes that we saw in electron microscopy, which is shown here. So this is the experimental data of the relative distribution of different sized filaments. So we ran the simulation using the concentrations we used in the actual experiment, and we saw very good agreement in the distribution of filament sizes. So we felt that that was a secondary way of validating this rate constants, or at least their ratio. Okay, so I wanted to focus on this rate constant here. If you notice, it's kind of slow. It's a second order rate constant. It's the assembly of 2 enzymes bound to DNA into the filament. And it's about 3 to 4 orders of magnitude slower than diffusion. So I thought that's kind of strange, because if the point was to speed up DNA cleavage, why would you put at the first steps a really slow rate constant? You know, it just seems strange to slow it down that way. But we thought this actually makes sense in terms of the biology. So this is a cartoon depiction of the cell. We have the genome of the bacterium shown here. There are a lot of primary sites, but they're invisible to our enzyme because they're methylated. There are many secondary sites. We're showing just one here, and we know our enzyme binds them tightly, but won't cleave them until it's drawn into a filament. And the only way to form a filament is to have primary sites around that can be bound by our enzyme SgrAI1, and now a filament can form between these species. We want this to be drawn into the filament, the secondary site bound to SgrAI1, because then it will be activated for cleavage, and more cleavage of a phage will facilitate its activation. We don't want to draw this one in because we don't want to damage our host DNA. This is where that really slow rate constant comes in. So if you calculate the rate of assembly of these 2 species, use the actual predicted concentration of DNA in the cell times our slow second order rate constant. Here's your rate. Now if we look here at the assembly of these 2 species, they're in much higher concentration than they actually are in the cell, and that's because they're tethered together. It's called the local concentration effect, and you can actually calculate it and estimate a concentration that it's experiencing. Multiply that by our slow rate constant, and we get a pretty fast rate, so 5 orders of magnitude faster. So assembly of this enzyme bound to secondary site is 100,000 times faster than assembly of this enzyme bound to secondary site. So we see filament formation on the phage and DNA cleavage, and we protected our genome. Okay, so that's great, but you don't need a filament model to do that. You can have something, just a binary model, and have that slow association rate constant. And here we ran a simulation with such a model and the same rate constants that we derived from our global fitting, and predicted DNA cleavage on the phage DNA shown by the solid line here. So that's cleavage of these sites, and then cleavage on the host DNA down here. So you see this sequestering of activity works perfectly fine with a binary model. So why a filament? So when we run the simulation with the filament, we see that DNA is cleaved much more rapidly. We still get good sequestering of activity. We still get very low predicted cleavage on the host, but the filament model is much faster. The filament mechanism is much faster. And part of the reason is that every time 2 species associate, there are two choices basically. So multiple ways to come together. there's also no competition. So you can imagine if it just came together, into a system of two, then these two on primary site would preferentially assemble and leave out the secondary site. So we don't have competition between species in the filament model. Okay, so you might say, well, if you want it to go faster, why not just ramp up this slow second order rate constant? So we did that computationally. We sped it up. It turned out it had to be about 3 times faster for the overall predicted rate of DNA cleavage to be the same as for our filament model. But now you see there's a lot more cleavage on the host DNA, because what was separating the activity on the phage DNA and the host DNA was this very slow rate constant, and having it sped up means we lose that sequestration. So our conclusion from this study was that the filament forming mechanism of SgrAI1 allows for sequestration of the secondary site activity onto the invading DNA, so we get more cleavages on the invading DNA, but we protect the host. And it's also more rapid than a binary mechanism, so the filament mechanism kind of makes up for the slow speed you end up with to have the sequestration effect. So that's my talk, and I want to acknowledge graduate students and undergraduates that participated in this work, and our EM collaborators, Dimitri Lomkis, Marajit Poli, our current collaborators, and Bridget Carragher and Clint Potter have been original collaborators on this, and thank you. Okay. So I'll take your questions now. So how do primary and secondary site decide association and dissociation of enzyme? Yeah, so that's a subject that I'm going to talk about tomorrow, is how this change in one base pair changes the activity of the enzyme so much. So my working model is that when the enzyme is bound to the secondary site, it's energetically more favorable to stay in a low activity conformation than when it's bound to primary site. So when it's bound to primary site, energetically, the protein DNA complex is more likely to be in a high activity state that's prone to filament. But when it's bound to secondary site, it's more energetically favorable to be in the low activity state that does not filament, but it's not so unfavorable that it can't be drawn into a filament if there's one there. So that's our working model. Another question, how many enzyme molecules are within micron-scale puncta cells? And how do you think that the linear filaments come together? That's actually a really, really important question. And I don't have that data. For my enzyme, I can probably estimate it, given the number of recognition sites on a phage DNA, that would limit the size of my filament. But in other enzymes, obviously, they form these much larger assemblies that must have many, many copies of the enzyme in them. And actually how the linear filaments come together in these, we don't know in most cases. I think I found one EM study where they could see the filaments were all aligned. And I think that makes sense in the case of an enzyme that gets inactivated by forming a filament, because then, it's all bundled together and perhaps protected from oxidation or degradation. So we have another question, how homogeneous is the filament? Is there evidence of branching fragmentation? In our EM study, we don't see any branching or fragmentation. We see this fast association, dissociation going on. And so the filaments are largely smaller, and you can get them to grow larger by ramping up the concentration. Okay, so I don't know how many minutes I'm in here. Do I take it into account of binding models between the protein subunits like induced fit, conformational selection? I think it's the conformational selection that determines the slow second order rate constant. So like I said, my model is that there's an equilibrium between a low and a high activity state. And the fraction of time it spends in the high activity state kind of speeds up the rate of filamentation. That second order rate constant is so slow because the enzyme is mostly in this low activity conformation that is not prone to filament. So it's only when it flickers in the high activity conformation that it's prone to filament. And filaments increase or decrease size depending on physiological conditions like starving. We don't have evidence of that. We haven't done a lot of in-cell studies. Most of our studies are in vitro. But that's an interesting question because the metabolic enzymes, many of them are actually affected by this. Do inactive mutants poison the rest of the filament? So inactive and cleaving DNA, I would imagine not, because even the cleaved DNA can assemble and induce filamentation. So the cleaved primary site is actually also an activator of filamentation and enzyme activity. But we think the secondary sites may poison the filament, so we're looking into that question now. Okay, so is the filamentation of enzymes influenced by the solvent effects such as molecular crowding? We haven't done that test, but I imagine it would be, but we haven't done that. It's a good question. And what prevents filaments to get crystallized? Okay, so there are some proteins, filaments that have been crystallized. There's a few I can think of, but usually when you try to make a crystal, you want like everything to be homogeneous. So you don't want an equilibrium between things of different sizes. That tends to poison a crystal. So that's why I think it's hard. I mean, we did try to crystallize those filaments, but never were successful. So we went with the M. So I'm not sure how I'm doing on time here. Okay, time for the next speaker. Great, thank you for all your great questions. So our next speaker is Tal Levin from the Weizman Institute. So he'll be speaking about exploring structural determinants and functional aspects of micron-scale protein assemblies in living cells.
Hello, thanks for inviting me. So I'll start. Like Nancy said, I'm Tal Levin from the Weizman Institute of Science. I'm a PhD student in the group of Emmanuel Levy. So to begin, we'll look at the possible effects on mutations on protein structure. We'll take this example of this homodimer. Mutations could either be neutral and don't affect the structure or almost don't affect it. Mutations in the core of the protein might induce misfolding of the protein and then aggregation of misfolded proteins. However, another possibility is that a mutation on the surface of the protein could induce new self-interaction. In this dimer, both mutations, the mutation is heightened by the effect of the protein being a dimer. So one mutation appears twice in the structure. In this case, both are on the same sides and could lead this dimer to form a tetramer in a finite assembly. Let's take another case that this time these mutations are on opposite sides of the dimer. So this time the new interaction size is not finite and it can continue infinitely. That's why we term this process agglomeration, because similar to aggregation, it could be infinite, but however, like oligomerization, the protein is still in its native state. To test, for example, agglomeration can appear in diseases like the case of sickle cell anemia, where a single mutation in hemoglobin from glutamate to valine now causes hemoglobin to form fibers and filaments or to agglomerate, and this actually changes the structure of the red blood cell, causing the disease phenotypes. Several reviews about agglomeration is what Nancy mentioned before, comprehensive lists of proteins known to agglomerate or cause filaments, and a review from our labs enhancing the effect of symmetry on the ability to agglomerate. In 2017, Hector Garcia, a postdoc from our group, showed that symmetric homomers are especially prone to form agglomeration. He did so by mutating 1 to 3 residues on the surface of symmetric homomers. These proteins were taken from E. coli and were bound to YFP, and then were introduced into wild-type yeast, and the fluorescent phenotype was then seen in the yeast. As you can see here, for these 12 proteins, in red, you see the sites of mutations. You see it multiple times due to the symmetry. And surprisingly, in all 12s, while the wild type had a cytosolic phenotype, the mutants showed either puncta or a filament, a fiber showing agglomeration, was very prevalent even with a few mutations. However, all of these mutations were 2 hydrophobic residues, either a tyrosine or a leucine, and thus we don't know the full effects of mutations to all residues and how they can impact the possibility to agglomerate. Here we see one of these proteins. We call it 1POC by its PDB entry code. It's a dipeptidase from E. coli. In light gray, we see 1 subunit, and in red, the 3 residues that were mutated. Like we said before, in order to see the effect of mutations to all different residues, we took 1POC and 1M3U and mutated them to many different amino acids, where each mutant had a different set of mutations in the 3 residues that were mutated in the 2017 paper. And like we can see here, some mutations had cytosolic phenotypes, other fiber, some had puncta, and some had nuclear phenotypes. We quantified it automatically using our algorithm, which marks regions of increased intensity inside the yeast. And then according to different parameters like aspect ratio and area, we automatically classify it to fibers, foci, nuclear, or condensates, which is both fibers and foci. For these 2 proteins, 1POC and 1M3U, we see that many different mutations were able to induce fiber or puncta formation. In 1POC, we have here the number of different mutants, each depicting a different trio of amino acids. More than 20% were forming fibers. Here, each column represents a different mutant. And surprisingly, 1M3U, more than 60% of them showed fibers. Seeing this was very prevalent. So now we wanted to see how different properties of the residues mutated might explain the phenotypes we see. So here on the left, we have stickiness, which is a metric somewhat like hydrophobicity, which means the propensity of a residue to be in an interface between proteins, rather to its ability to be, the propensity to be on the surface. So we see here in gray the cytosolic, and in red, the fibers plus the foci. And we see that for 1POC, the more sticky the residues that we mutated, we saw more fibers and foci, meaning that we needed a positive selection to induce fiber formation. And similarly, we see here that the charge and neutral charge showed more fibers and foci, while either negative or positive, we had more cytosolic phenotype and reduced fiber and foci in vivo. And on the contrary, 1M3U showed a very different picture. No matter the stickiness of the residues, it had a very high percentage of fibers and foci. But in the charge, what we see that a very negative charge was able to dissolve, to keep the protein from cytosolic, not allowing to form fibers and foci. This shows that in contrary to 1POC, 1M3U requires a negative selection, or the wild type acting as gatekeepers, preventing it from agglomeration. All of these proteins were proteins from E. coli in yeast. We wanted now to take a look at the yeast proteins. Here also we see, just a side note, 1POC and 1M3U, neutral mutations to 3 glycines and 3 alanines. We see that in 1POC, the phenotype is cytosolic, while 1M3U, it's fiber, again strengthening the fact that 1POC requires positive design and 1M3U a negative design. Now we wanted to see natural yeast proteins, like those shown in large-scale screens, how the agglomeration in these proteins affect the phenotypes of the cells. What we did, using structural properties alone, we decided residues that we might think that mutating them would reduce agglomeration. We did this blindly, strictly based on structural properties. And you can see in red the sites mutated for each protein. And for 3 of them, ACC1, PRS5, and ASN2, these mutations were able to significantly reduce the amount of agglomeration we see, both fiber and puncta. And now with these non-agglomerating mutants, we wanted to see how the fact that now these proteins are cytosolic would impact the cellular fitness. The first experiment we did was a simple growth assay. We had cells in stationary phase, allowed them to grow, and we measured 2 main parameters. The first, the lag phase, the time since moving to a new media until the cells reach their peak exponential growth rate, and the maximal growth rate that we actually saw. While we didn't see any difference between the maximal growth rates of the wild type to the non-agglomerating mutants, we did see an increase in the lag phase, with the mutants that do not agglomerate are spent more time in the log phase, with ACC1 having a much larger gap than both PRS5 and ASN2. Now we wondered what causes this difference. Thought maybe it's the fact that cells could die if they don't have these agglomerates. So in order to do this, we checked death assays, where we took both the wild type and the mutants and kept them in stationary phase and in the same media. And each day we took a same volume imaged after staining for dead cells. We see here that in ACC1, there is a big difference between the wild type and the mutant, both in the beginning and over time. We see much more dead cells in the non-agglomerating mutant. On the contrary, both in PRS5 and ASN2, we see very similar pictures between the agglomerating and non-agglomerating mutants. So to summary of the first project, we see that some proteins require positive design in order to agglomerate, while others, like 1M3U, require a negative design to remain soluble. With any small mutation would induce agglomeration. In the second project, we see that natural agglomerates impact both lag phase duration and some impact cellular survival in stationary phase. Note that this is still a work in progress, and I would appreciate any feedback. I would like to thank my lab. I would like to thank Emmanuel, the principal investigator, to Hector, who did the experiments with me, and to Charlie, who helped design the automatic phenotypes. Thank you.
That's great. Thank you very much. So I'll take questions now. Okay, so we have our first question. Does the oligomerization state of native proteins predict how likely they are to agglomerate upon mutation? i.e. octamers are more likely than tetramers, are more likely than dimers, etc.
In the first experiment that I showed, the one from 2017, all proteins were D4 and D5 symmetries. And what we see in the second project, in the wild type, we have very different symmetric states. For example, ICC1, which is a C2 symmetry. This is something that we want to explore and are currently thinking about how to do this and actually check the different symmetry states.
Great. Yeah, I really like this paper because it showed how relatively easy it is to mutate any protein to form a filament. So it's not like, you know, evolving a filamentous form would be some huge burden to nature to do. It's actually can take maybe just one amino acid change in the right place.
Yeah.
Another question, it may be useful to check if the visible puncta and the agglomerate mutants are sequestration of the protein due to a protein quality control response, or if they are a phase of the protein.
That is a good question. What we see that in these cases, the non-agglomerating, both in the wild type protein, we see the phenotype mainly is cytosolic. In the proteins that in the 2017 papers that were not mutated, they were cytosolic. It's true that puncta could be protein quality control, but it's something that we will address in future work.
Okay, another question, is there any bioinformatic bias about the functions of proteins that are either positively or negatively affected?
We wanted to try to look specifically at structural metrics without looking at exactly how the protein forms a fiber. So there are biases. For example, we want to try, we have the axis of symmetry, which is an important, we assume that fibers would form along the axis of symmetry, for example. So this is a sort of bias.
No bias towards function of the given protein?
The functions of the protein, we try to do it relatively blindly. For example, for the proteins in yeast that are known to form fibers, we just chose the proteins that are known. There is a limited set that we could experimentally verify, so we chose those.
Okay, great. So there's a few more questions. You can type your answer. We have to move on to the next speaker. Thank you, Tal. The next speaker is James Wilhelm from University of California, San Diego. So I actually showed one of his papers. So it was one of the early papers that showed this self-association using screens. So his title is Identification of an Actin Fold Containing Metabolic Enzyme that Undergoes Regulated Polymerization.
Thanks, Nancy, and thanks to the organizers for inviting me to speak today and in this kind of interesting situation. So yeah, the first two talks were great in terms of a lead, and I'm glad I deleted a bunch of slides before coming here because they gave a lot of the background. So I'd like to start with the more recent screen that just came out in 2019, which is where we screened all 440 metabolic enzymes in the yeast GFP strain collection for the ability to form structures under 3 different growth conditions. And we found 60 of the metabolic enzymes will form either filaments or some other type of structure in more than 10% of the cells. And so that's given us a huge number of targets to play with, which obviously leads to a question of which ones do you prioritize. And so one of the enzymes that came out of that screen polymerized, assembled under really unusual conditions. It was the only one that assembled in log phase specifically, and that was the enzyme glucokinase. And just to orient you to all of metabolism, the little circle is glucose, and then the line is glycolysis leading to citric acid cycle. So glucokinase is the first committed step in glycolysis. And the reason we were interested in it was because our longstanding interest in the cytoskeleton. So there's been a long history in terms of understanding the structures of sugar kinases, and they are part of the actin superfamily. So hexokinases have an actin fold and have similar conformational changes to actin, in that in an open conformation, they bind ATP and glucose. That triggers a closed conformation that leads to hydrolysis of the ATP and phosphotransfer. And then they release the products and they recycle back to the open conformation. And so this has a lot of similarities to what goes on in terms of the ATP hydrolysis cycle in actin. And it's long been hypothesized, going back to when these structures were first determined, that perhaps actin evolved from one of the sugar kinases. And in order to do that, as the previous speaker noted, what you need is a conformation-specific binding to self. So you need the enzyme to undergo self-assembly when bound to its substrates, but then at some point in evolution lose perhaps the sugar binding, but retain the ATP binding and hydrolysis. So this has been a long-standing idea. And given the ease with which proteins can polymerize, the question was, well, if it's so easy to make all these polymers, why haven't we found something from that's structurally analogous or structurally related to actin that can undergo regulated polymerization? So finding glucokinase in our screen was particularly interesting. So we began to investigate its polymerization properties. And shown here are the results of an in vitro pelleting assay. And so what we found is that protein alone, ATP, or glucose individually generated very little pelleting, but if you combine both substrates, ATP and glucose, you get a substantial amount of pelleting. Now, of course, with all these proteins, the issue is it actually forming a filament or is it just aggregating? And so we validated that using negative stain EM, and as you can see here, with glucose and ATP, you see filaments forming. Glucose triggers a small amount of just aggregation, and ATP alone causes nothing. So in terms of where the questions we're interested in, interested in investigating, one was, is ATP hydrolysis required for regulating glucokinase assembly? Do the products regulate the assembly? Because end product inhibition is often a trigger for a number of these enzymes. And tying back the in vitro work back to what the in vivo trigger might be. So is ATP hydrolysis required to assemble a glucokinase filament? It's not, and so we showed that in a number of different ways. But basically, using a variety of non-hydrolyzable analogs, you can see that we only got pelleting when both substrates were present. And so if glucose in the presence of either a non-hydrolyzable ATP, such as AMP, PMP, or ATP gamma S, we got polymerization, although it was less than what we see with ATP and glucose. And just to show you what these filaments look like, they, again, are forming filaments. Now, one of the reviewers on our paper was concerned about the low level of hydrolysis of these non-hydrolyzable analogs. So we've also done this with 6-deoxyglucose, which can bind to glucokinase, but can't receive the phosphate upon hydrolysis. Is ATP hydrolysis required for glucokinase assembly? The answer is no. And you might think that that's where the story ends, because why would you have something regulated by substrate binding and product binding? But in fact, this enzyme is unusual in that ATP and glucose together can trigger polymerization. And as I showed you, having non-hydrolyzable analogs will also generate the polymer. But interestingly, glucose 6-phosphate on its own will also trigger pelleting, and it also triggers polymerization, as shown here. So here's the glucose ATP filaments, and here's the glucose 6-phosphate filaments. They're slightly different in terms of their thickness, but the polymerization is quite robust. Now, what was particularly striking, got us very excited, was that if you combine both products together, so the products of the reaction are ADP and glucose 6-phosphate. Glucose 6-phosphate on its own will trigger polymerization. But if we then add ADP to the glucose 6-phosphate filaments, we can rapidly disassemble them. So what we were seeing was that the substrate binding could trigger polymerization. The binding of one of the products could trigger polymerization, but the presence of both products inhibited polymerization. In fact, adding the second product could actively disassemble the glucose 6-phosphate filament. So that suggested perhaps that maybe we were looking at two distinct filaments. And so we were able to collaborate with Mark Herzig and take our negative stain data and do a 3-dimensional reconstruction to 20 angstroms of both the glucose 6-phosphate filament and the ATP glucose filament. And at 20 angstroms, they have the same basic structure. And this structure is also very similar to the recently published Stoddard structure from the Coleman, Garner, and Murray labs. And so at gross level, they're not distinct filaments, but that doesn't rule out that there's some interesting things happening at the subunit interface. So these filaments are quite odd in that they are regulated by both their substrates and their products, but the relative amounts of the products can have radically different effects on assembly. So that raised the question of, well, what is the actual in vivo trigger for polymerization? Is it the substrates, one or both of the products? What's the role of these things going forward? Now, this is actually proven to be a really difficult problem because, as you may have noticed, that even though I started with a GFP protein screen, I haven't shown you the GFP tagged protein. And that's because what we found is that this protein is incredibly sensitive to the modifications to the C-terminus. And so adding tags to the C-terminus actually hypersensitizes it to polymerization. So while it came out as a positive hit in our screen, and it clearly can polymerize, the regulation of the polymerization is drastically altered by GFP or HA tags or other kinds of tags. So we had to go back and develop new ways to kind of basically use immunofluorescence to track what the actual in vivo triggers are. And so what we started with was examining the question of whether glucose 6-phosphate is the trigger in vivo as it is in vitro. And so here's glycolysis, the bane of many undergraduates' existence. And glucokinase acts at the first step. And what I'm going to focus on are mutations in phosphoglucoisomerase and phosphoglycerate kinase. The idea being that if we mutate different steps in glycolysis, we can generate different levels of glucose 6-phosphate and perhaps trigger polymerization. So if we mutate the next step in glycolysis, we should expect a buildup of glucose 6-phosphate that in theory, if glucose 6-phosphate is the in vivo trigger, we should see the unmodified protein polymerize in vivo. And that's exactly what we see. And so without adding fresh glucose to the mutant strain, we only see a small amount of low-level self-assembly. But when we add fresh glucose, we see full-blown filaments. And these are not associating with actin, microtubules, they're not in mitochondria. They seem to be autonomous structures as far as we can tell. Now, are these structures aggregates or are they reversible? It turns out that we can add fructose and bypass the block step and reactivate metabolism. And when we do that, we can actually so if we add fructose to strains where filaments have been induced, we can actually trigger disassembly. So, and furthermore, this gain and loss of filaments is not due to changes in protein level, expression level. It seems to be, or it's not protein degradation, it's actual reversal of the polymerization in vivo. So you could imagine two models. One, it's the buildup of glucose 6-phosphate triggering assembly, or that we've just lobbed a grenade into glycolysis, and any disruption of glycolysis would trigger assembly. So the idea would be if we block glycolysis at a downstream step, it will allow us to distinguish between these two models because mutations that block lower in, further down in glycolysis should accumulate less glucose 6-phosphate. So if we mutate phosphoglycerate kinase and block this downstream step, we would expect it to have significantly less glucokinase assembly than if we blocked the PGI1 step. And that's exactly what we see. So if you mutate downstream steps in glycolysis, you get less and less assembly. So, and we've done a whole gradient of these mutations. So the closer the block is to glucokinase's step, the more assembly we see. So we think this is pretty good evidence that glucose 6-phosphate triggers polymerization in vivo. It doesn't rule out that ATP and glucose can do it. But given that we're focused on the native protein, it looks like glucose 6-phosphate, at least on its own, or buildup, asymmetric buildup of glucose 6-phosphate can trigger glucokinase polymerization in vivo. So, so our model is that at least in vivo, glucose 6-phosphate is a trigger for assembly in vivo and in vitro. And what I'd like to end with is kind of relating our data back to implications about thinking about the evolution of actin. Now, these glucokinase filaments are structurally very different from actin filaments, but the data I've shown you has some striking similarities to the things that we know about how actin hydrolysis drives dynamic filament assembly. So both filaments are sensitive to the state of the gamma phosphate in the ATP. So for actin, ATP actin will polymerize, the filament is enzymatically active, and that loss of the gamma phosphate massively destabilizes the filament, and you lose ADP actin off the opposite end. Now, we don't know that the filaments are enzymatically active. In fact, we suspect that they aren't, but if you were to build a glucokinase filament that was enzymatically active, you could, in theory, generate very similar types of dynamics where an ATP glucose subunit bound and was stably forming filaments, and then the intrinsic enzyme activity would generate an ADP glucose 6-phosphate state. And we know that that doesn't support filament assembly, and it would fall apart. And so we've actively been exploring this, and I don't have time to get into that, but it suggests a path for making glucokinase filaments dynamic like actin. And we've actually made a fair amount of progress on this before pandemic shut down and everything. So, so with that, I'd just like to thank funding. And Michael Carver is an undergraduate who kind of took up a lot of the biochemistry and has really driven this project forward. And particularly Mark Kurtzig, who's been working with Michael on the cryo-EM reconstruction of the glucose 6-phosphate filaments. So with that, I can open it up to questions.
Okay, great. Thank you for that great talk. So we have a couple questions here. One says, very nice. The fluorescence images showing lack of assembly actually show a granulated pattern. These granules may respond to small filaments, suggesting that there is assembly but not long polymerization. Is this correct? So that there's small filaments in the granulated foci.
Yeah, I mean, I think that's always possible. I think what I would say is that it doesn't mean that it's not forming. It's that there's always some of that is very hard to distinguish because it's immunofluorescence versus, normally you'd like to work with a live image, and so that everything always looks slightly granulated with immunofluorescence. But I agree. That's why there is a low level. That's why we quantitate it. There is a low level of assembly. So.
Okay, great. So I think we have to move on to the next speaker, but there are a lot of questions for you. And so there's a way to type in your answer. So you can, thank you very much. So our next speaker is Weihan Li from Albert Einstein College of Medicine, entitled Regulating ER Protein Homeostasis by Differentially Processing mRNAs.
Hey, perfect. Great, thank you. First, I'd like to thank you for your kind invitation. Let's see, just starting there you go. Okay, first I'd like to thank for the kind invitation, and my name is Weihan. I'm currently a postdoc in Robert Singer's lab at Albert Einstein College of Medicine. And today I'd like to share with you the work I did when I was a PhD student in Peter Waters' lab at UCSF. So as you know, the endoplasmic reticulum, or ER, is the major folding compartment. And for most of the secretory proteins and transmembrane proteins, they're folded in the ER. So these proteins enter ER as these unfolded proteins, and then with the help of ER chaperones, they get properly folded and then packaged and delivered. So within the ER, there's this balance between the ER folding capacity and ER folding burden. Namely, there has to be enough ER and ER chaperone to fold these unfolded proteins. However, this balance may be disrupted under certain circumstances, causing exceeding unfolded proteins, accumulation of unfolded proteins in the ER, which is cytotoxic. But fortunately, cells evolve a collective of signaling pathways termed the unfolded protein response, which can alleviate the ER stress and restore the ER protein folding homeostasis. And for today's talk, I'll focus on the most evolutionarily conserved branch of the unfolded protein response, which is mediated by this protein called IRE1. So I'll zoom into IRE1. IRE1 is this ER transmembrane protein. It has a ER luminal domain and a cytoplasmic domain. Upon accumulation of unfolded proteins, the luminal domain of IRE1 can recognize these unfolded proteins and trigger dimerization and oligomerization. And subsequently, the oligomerization and dimerization would activate the cytoplasmic RNase domain of the IRE1. And the activated RNase domain can very specifically cleave at 2 positions on a specific mRNA called XBP1 mRNA. And the cleavage will remove the intron, and then the 2 exons can be ligated to form a spliced XBP1 mRNA. The spliced XBP1 mRNA produces the XBP1 protein, which is a transcriptional factor that can transcriptionally increase the ER chaperone, increase ER volume, and so on and so forth, so that the ER folding capacity is increased and the ER homeostasis can be restored. And so this is the first functional output of mammalian IRE1. And about 15 years ago, people found a second functional output for mammalian IRE1. They found that the mammalian IRE1 can decrease the ER folding burden by selectively cleave a subset of mRNAs at the ER periphery. Just want to remind you that the mRNAs that encode ER targeted proteins are translated at the ER periphery so that the newly synthesized proteins can be co-translationally deposited into the ER lumen. And upon ER stress, the activated IRE1 can cleave a subset of the ER peripheral mRNAs, and the cleavage products are subject to further degradation by housekeeping machineries. So by cleaving and downregulating the mRNAs that encode the ER unfolded proteins, IRE1 can very efficiently decrease the folding burden that enters the ER and restore the homeostasis. So this is the second functional output of mammalian IRE1. So we know that there are 2 very distinct mechanisms for IRE1 to alleviate ER stress, namely the non-conventional mRNA splicing and selective mRNA decay. And it has been a standing question in the field to understand what is the molecular distinction between the 2 functional outputs. So what determines non-conventional mRNA splicing versus selective mRNA decay? And it's hard to answer that question in mammalian cells because the 2 functional outputs are mixed or entangled in mammalian system. But fortunately, the nature have evolved 2 fantastic yeast species, Cerevisiae and Pombe, which provides a fantastic platform for this specific question. In Saccharomyces cerevisiae, the Cerevisiae IRE1 homolog only has 1 functional output, which is the non-conventional mRNA splicing, but it doesn't do the selective mRNA decay. Vice versa, in contrast, for Saccharomyces pombe, the Pombe IRE1 only does the selective mRNA decay, but not the non-conventional mRNA splicing. So in Saccharomyces cerevisiae and Pombe and Saccharomyces pombe, the IRE1s are specialized to only one of the 2 functional outputs and allow us to address the question, what leads to the distinct functional outputs of IRE1? And to address that question, we purified the Cerevisiae and Pombe IRE1 homologs and tested in vitro. This is an in vitro assay with purified first Cerevisiae RNA incubated with Cerevisiae IRE1 over a time course. Top band is uncleaved, bottom band is cleavage product. As you can see, the Cerevisiae IRE1 can cleave its endogenous Cerevisiae RNA substrate. However, with Pombe IRE1 doesn't cleave the Cerevisiae RNA. Similarly, with Pombe RNA, cerevisiator-1 doesn't cleave the Pombe RNA substrate, but the Pombe RNA-1 can cleave its cognate RNA substrate in vitro. So this in vitro assays showed that the two enzymes, cerevisiator and Pombe, can distinctively recognize its own substrates in vitro and have distinct RNA specificity. And we further went on to characterize the motifs that are recognized by cerevisiator and Pombe. Long story short, we found that cerevisiator-1 is more stringent and recognizes a longer sequence motif, CNG, located on a 7-mer stem loop. The loop size is confined to 7. While Pombe RNA-1 is more promiscuous, recognizes a shorter sequence motif of UGC, located on a stem loop as well, but the loop size can be variable from 3-mer all the way to 9-mer. And we further tested this, this finding by designing this common substrate, which has the cerevisiator motif as well as the Pombe motif. And by prediction, it should be cleaved by both. And very nicely indeed, the common substrate is very efficiently cleaved by both cerevisiator-1 and Pombe RNA-1 in vitro, further validating our finding. So far I showed to you that the cerevisiator-1 and Pombe RNA-1 have distinct RNA specificity, but one main question is still unanswered is that what are the residues or what are the RNA-1 structural features determines the RNA specificity? And before diving into that question, I'd like to share a little story behind the scene with you, namely that when I was trying to purify the Pombe RNA-1, it was really tough protein to be purified. It took me over 2 years to purify this protein. It was a kind of a nightmare. And it turns out that the reason why it's so difficult to purify is that the Pombe RNA-1 is toxic to bacteria cells. And I'll show you what I mean by toxicity. So in the following assay, simply put RNA-1 cytosolic domain under a IPTG-induced growth promoter in bacteria cells, and then measure the growth curve. So here's the growth curve for bacteria cells that express harvest the empty plasmid or expressing a cerevisiator-1 cytosolic domain, it grows fine. But for the bacteria cells that expresses a Pombe RNA-1 cytosolic domain, it simply doesn't grow within this time window. Remember that I mentioned the Pombe RNA-1 is more promiscuous in terms of its RNA specificity. So we wonder if it's the promiscuous RNase activity of Pombe RNA-1 that causes the toxicity, for example, by cleaving a lot of endogenous bacterial RNA. And so to test this idea, we simply added a RNase inhibitor of RNA-1 that has been previously characterized into the bacterial culture. So we just add the drug into the bacterial culture. And very nicely, the inhibitor, the RNA-1 RNase inhibitor, very nicely reverted the toxic effect. And the bacteria is no longer dead. It's started to grow. So this assay turned out to be very powerful in terms of examining the promiscuity of an RNA-1. Because, for example, if an RNA-1 is toxic, like Pombe, it suggests that the RNA-1 has a promiscuous RNase. And if RNA-1 is not toxic, like cerevisiator-1, then it has a stringent RNase. So we serendipitously turned a nightmare of protein purification into a dream assay to test the RNase specificity. And we used that assay to address whether can we make cerevisiator-1 Pombe-like? Can we make cerevisiator-1 more promiscuous, just like Pombe, so that we can know which structure determines the promiscuity. And to this end, long story short, we systematically examined the differences, all the different residues between cerevisiator and Pombe RNA-1 RNase domain, and we identified 3 residues. In the following experiment, we used cerevisiator-1 as a backbone and changed these 3 residues into the Pombe corresponding Pombe counterparts, and creating a construct, we call it mut3, cerevisiator-1 mut3. We put it into the test of bacteria assay, and as I show you, cerevisiator-1 wild type is not toxic, wild type Pombe is toxic, and then the cerevisiator-1 mut3 is toxic. By adding the inhibitor of RNase inhibitor of RNA-1, the toxicity goes away. We further validated this finding by doing the in vitro cleavage assay. Pombe RNA is not cleaved by wild type cerevisiator-1. And then with the mut3 can very efficiently cleave this Pombe RNA now. actually we further characterized the 3 mutations. We found that you don't need all the 3, you just need 2 of them. So with the cerevisiae mut2, the mut2 is already becoming more promiscuous and can cleave the polymer RNA already. So where are the 2 mutations on IRA1? This is a cartoon illustration of a polymer IRA1 dimer. So this is protomer 1, protomer 2, form a dimer, and down here is the IRA1 RNA cleavage, interaction site and cleavage, where the cleavage occurs. Surprisingly, the 2 mutations are not located on the IRA1 RNA interface. Instead, the two mutations are located at the IRA1 dimer interface. More specifically, this is the residue, protomer 1, protomer 2. The green residues, lysine, tyrosine, are the 2 identified residues. In the wild type, there's an intermolecular salt bridge across the dimer interface. And when the 2 mutations are made, so this is a lysine to aspartate and tyrosine to arginine, we found that, of course, the original salt bridge is disrupted and is lost, no longer there. And by using a combination of computational and biochemical method, we found that a new pair of salt bridges formed between the 2 identified mutation residues between aspartate and arginine. And we found that this rearrangement at the dimer interface causes a structural rearrangement at the dimer interface, as well as and the structure rearrangement propagates to the IRA1 RNA interaction interface. And we think this structural rearrangement causes the RNA specificity change that we have observed previously. So with that, I'd like to thank the whole Water Lab, especially Peter, for being a great mentor. And this movie is one of the most memorable moments of my PhD life, when we pushed Peter into the swimming pool during one of the lab retreat. And of course, I'd like to thank Callie, Wojtek, Yurka, Philip, Carlos for the wonderful work, that collaboration we did, and also Diego from Matt Jacobson's lab that we collaborated on the MD simulation of the mutant IRA1. And of course, my current postdoc advisor, Rob Singer, for providing a new academic home for me. And with that, I'd like to thank you for your attention, and I'm happy to take any questions.
Great, thank you for that very interesting talk. So this enzyme IRE1 is known to form filaments, right? So
yeah.
Where does that fit into these activities?
So the monomer IRA1 is not active, and only when IRA1 is forming either a dimer or a oligomer, it becomes activated.
So is there a difference in activity? I thought there was some data that seemed to suggest the non-specific activity, the promiscuous activity, might be the polymeric form, the ligament, and the dimer form responsible for the specific cleavage sites.
Yes, there are some studies suggest that maybe a higher order oligomers is having a more stringent RNase activity, while a lower order oligomer like dimers have a more promiscuous activity. And also vice versa. There's different studies suggesting.
Yeah, I remember there being a lot out there on this and that being a little confusing. So, okay.
Yeah.
Go ahead.
Yeah, but still it's not clear at this moment whether there are different ways to regulate the RNA specificity. One way is probably the oligomeric state, and others are probably, like, say, the phosphorylation status or the protomer alignment indicated in this study. So yes, definitely the oligomeric state may be one of the ways to regulate.
Okay, so we have a couple questions in the question and answer box. First from Juan Pedro Luna Arias. What is the phenotype of null IRE1 mutants in these 2 yeasts? Are there IRE1 orthologs in other systems like human and Drosophila? 2 questions.
So if you knock out IRE1, the cells will not grow under conditions where ER stress is induced. So if you cause unfolded proteins in the ER, if the cerevisial polymer doesn't have IRE1, it won't grow. And what is the second question? Yeah.
Are there orthologs in other systems like human and Drosophila?
So RE1 is evolutionarily conserved from yeast to human, and apart from cerevisiae and pombe, almost all the metazoan RE1 have both non-conventional MR splicing and selective MR decay.
Okay, great. So I think we have to move on. There's some questions that you can answer by typing in the Q&A box. So thank you very much.
Thanks.
Yeah. Okay, our next speaker is Gregory Fuller from Johns Hopkins University, and his talk is titled Glycolysis Enzymes Coalesce in G bodies in Hypoxia Nucleated by RNA.
Hi. All right. Thank you for the introduction, and thank you for giving me a chance to talk here today. I'm Greg Fuller. I'm a graduate student in John Kim's lab, and I'm going to tell you about how RNA can help organize glycolysis enzymes in yeast in response to hypoxic stress.
Presenter view, if you want to move it to.
Oh, thank you.
Or something.
So this project really started when a former graduate student in the lab, Ting Han, wanted to identify all of the RNA binding proteins in yeast, and he did this using interactome capture, where he incorporated 4-thiouracil into RNA, used UV light to cross-link RNAs and proteins, captured these with the poly-A tail, and used mass spectrometry to identify the RNA binding proteins. When he did this, he found a large number of RNA binding proteins in yeast, many with known RNA binding domains, but also a large subset that lack any known RNA binding domains. So these are non-canonical RNA binding proteins. Included within these were a large number of metabolic enzymes, including phosphofructokinase, aldolase, enolase, and pyruvate kinase, all enzymes of glycolysis. So we wanted to study RNA binding of the glycolysis enzymes in more detail. And to do this, we did a PAR clip where, again, we're cross-linking RNAs and proteins and capturing the RNAs associated with each specific protein. So what you're seeing here is autoradiography of labeled RNA that's co-migrating with protein that you can see blotted here on the right. And so each of these glycolysis enzymes has the ability to bind to RNA. When we sequenced these RNAs, we found that there are many RNAs specific to each glycolysis enzyme, but also a large subset of RNAs that are bound by multiple glycolysis enzymes. So we wanted to know if there were any conditions under which this RNA binding activity might modulate the localization of these enzymes. So we looked through a few conditions and we found that specifically under hypoxic stress, the glycolysis enzymes move from a diffuse localization to large single puncta. And this was true for multiple glycolysis enzymes. Next, we looked at whether or not they co-localize with each other. So we used a PFK2 azurite and looked at localization of PFK1 GFP, FBA1 GFP, and CDC19 GFP, and found that we could detect a perfect overlap with PFK2 azurite and these GFP tag glycolysis enzymes, suggesting that all of these enzymes are actually coalescing into singular structures. So we call these glycolytic bodies or G bodies. And the fact that this happened specifically in hypoxic stress was really interesting to us because it suggested a mechanism or a function of this coalescence. So under normal conditions, cells are using glycolysis to convert glucose to pyruvate and generating a little bit of ATP, and further processing this pyruvate through respiration to generate a lot of ATP. However, respiration is oxygen dependent, and in hypoxic conditions, you're only getting a small amount of ATP for every molecule of glucose. So we hypothesized that all of these enzymes are coalescing in single structures in order to enhance the rate of glycolysis and produce more energy for the cell. And so we cultured cells under two different conditions, either in hypoxia in order to form G bodies, or we left them in normoxia such that the enzymes were still diffuse. We then shifted these populations into fresh media and measured how quickly they could deplete glucose from the media. We found that the cells that had been in hypoxia and therefore had G-bodies were able to deplete glucose from the media at a faster rate than cells lacking G-bodies, suggesting that G-body formation is correlated with increased glucose consumption. So recently, phase separation has been shown to be a common mechanism that can facilitate reorganizing proteins and RNA. So we wanted to ask if G-bodies have any of the hallmarks of phase separation, such as the ability to fuse and dynamic exchange with the cytoplasm. So to test if these can fuse, we mated A and alpha cells, and I'm going to play you a video here in a moment of these mating cells that each have G-bodies. And first I want you to pay attention to this cell where these two granules are going to fuse, and then I want you to pay attention to this cell where these two granules are going to fuse. So very quickly, these two come into contact and fuse into one single structure. A little bit later, these two come into contact and over time fuse into one single granule. So it seems that G-bodies have the ability to fuse. Next, we did fluorescence recovery after photobleaching of two different G-body markers, FBA1 and PFK2-GFP. We found that over long time scales, tens of minutes, we could detect up to 20% recovery, suggesting that there's weak exchange between the granule and the cytoplasm. So that's not consistent with these kinds of liquid phase separations, but some more solid gel-like structure. So we wanted to determine what the components of G-bodies are. And we found that we could purify G-bodies using differential centrifugation followed by affinity capture. So what you're seeing here are actually dynabeads. And when we pull down a normoxyglycate, you don't see any kind of puncta accumulating on the dynabeads. However, when we take the hypoxyglycate, we're able to capture seemingly intact G-bodies along these beads. So we used mass spectrometry to identify proteins in G-bodies and validated this again with co-localization with PFK2 azurite. And these are some examples of the co-localized proteins. So when we looked at the G-body proteome, we found proteins involved in a large number of cell processes, including protein chaperones, proteins from other metabolic pathways, and proteins from the proteasome. But the big unifying feature was that almost all of these proteins are non-canonical RNA binding proteins, raising the possibility that G-bodies are RNP granules. So we wanted to purify RNAs from G-bodies to characterize RNAs in these granules. And so we used the same purification procedure, only this time extracted RNA and sequenced the G-body associated RNAs. By comparing the RNAs that came through in the flow through to the RNAs co-purifying with G-bodies, we were able to determine a subset of RNAs that we deemed to be high confidence G-body RNAs. So we took several of these and did SM fish with a PFK2-GFP marker to test if these RNAs are associating with G-bodies. And for each RNA we tested, we detected cases of overlap, while most of the RNA is outside of the granule. We also frequently observed this kind of peripheral localization pattern where the RNA surrounds the granule. When we did this for a number of different RNAs and plotted the fraction of the RNA that co-localizes with the granule against the enrichment we determined through RNA sequencing, we saw a nice correlation validating our RNA sequencing approach. So we compared the fraction of mRNAs bound by glycolysis enzymes in normoxia through Parclip in the transcriptome to the set of G-body enriched RNAs. And we found that the G-body enriched RNAs were, or that the normoxic bound RNAs were nearly twofold enriched in the set of RNAs found in G-bodies. Suggesting that the same RNAs that are bound by glycolysis enzymes in normoxia are going to G-bodies in hypoxia, raising the possibility that these RNAs are helping to bring these enzymes together into these granules. So we wanted to specifically test the role of RNA, both in G-body structure, as well as the role of RNA in forming these granules. To accomplish this, we made a fusion protein of PFK2, which strongly localizes to G-bodies, with MQSR, a non-specific endoribonuclease. And we put this under an inducible promoter. So when we add copper sulfate, we're able to cause expression of this fusion protein. What we found was that as we increase expression of the fusion protein, we decrease G-body formation. Whereas in vector control cells, there's no difference in G-body formation regardless of the concentration of copper added, suggesting that RNA is required for G-body formation. Next, we wanted to ask what the role of RNA is in these existing structures. So we cultured cells with a plasmid expressing the fusion protein and just vector control. And shifted these cells to hypoxia without any induction such that they would each form granules. And then we induced expression and shifted the cells to normoxic conditions to ask what happens when we target an RNase to an existing G-body. What we found is that in vector control cells, you either have single puncta or no puncta whatsoever after their shift to normoxic conditions. However, in the cells expressing the fusion protein, we would frequently observe multiple foci scattered throughout the cell. That's quantified here on the right. So we never observe these multiple foci in the vector control cells. So this suggests that targeting the nuclease to the G-body caused it to fracture into multiple pieces. Ultimately, we propose a model where under normoxic conditions, these glycolysis enzymes are diffused throughout the cytoplasm and sometimes bound to RNA. When we shift to hypoxic conditions, these RNAs provide multiple binding sites to bring these glycolysis enzymes together. And over time, these accumulate more glycolysis enzymes in RNA, and through fusion and this accumulation process, ultimately form a mature G-body. With that, I would just like to thank everyone in the lab, John Kim, a great mentor, everyone who's worked on the G-body project, so Amelia, Alex, Joy, Ting, and Mallory, as well as our collaborators in the Yates lab who helped us with our mass spectrometry, and the Mian lab who helped us with our FRAP experiments, and in the Mayan Jin in the Kliansky lab who did a lot of the initial characterization of G-bodies. And with that, I'll take any questions. Thank you.
Great, thank you very much. That was really fascinating. RNA, so it's the RNA that's kind of scaffolding things to bring it into one G-body instead of multiple ones. And so having them all together in one has some advantage, you think?
Yeah, that's what we think is going on. We're hoping to test that more mechanistically in the future since we seem to be able to purify these.
So this is a case of upregulation by forming these kind of self-assemblies. Really interesting. Yeah. I was curious, you know, I always hear about the metabolic enzymes are at relatively high concentrations in the cell and often contaminate pull downs and that sort of thing. But, you know, if that was a possibility with the RNA pull downs, but with all the data together, it looks pretty compelling.
Thanks.
All right, questions from, oh, okay. Let's see. We have a lot of, okay, here. So Simone Curry, Gregory, great talk. How are you thinking about the fish results showing RNA largely outside of the G bodies in terms of your model?
Yeah, we don't think that the majority of any given species of RNA is recruited to a G body, that there just has to be some RNA there to help bring these enzymes together. So I think there's multiple protein-protein interactions that are likely happening, but it seems that RNA has to be there to initially start them coming together.
Okay, we have Emmanuel Levy. Beautiful. Did you also observe an enrichment in mRNA encoding the proteins of the G bodies, like PFK and enolase?
Yeah, we have found that in normoxia, the RNAs bound by glycolysis enzymes include the mRNAs for the other glycolysis enzymes. That enrichment was smaller in hypoxia, but yeah, these enzymes seem to bind their own mRNAs.
That would decrease translation, one would think. Just interesting. Okay, Shahram Venkatesan, great talk, Gregory. Do we know if these glycolysis enzymes increase in activity when the G bodies, than when diffuse? So is the activity upregulated in the G body relative to when it's diffuse? Do we know this?
We don't know the answer to that question yet, and we're really hoping to test that as soon as we can get back in the lab.
But that's like your model, your hypothesis.
Yeah, that's our model. We just don't know the answer yet, though.
That's great. Okay, so we have a few more questions in the Q&A box, and due to time, we have to move on. Thank you again.
Okay, thank you.
Okay, now we have Eric Lynch from University of Washington with his talk entitled Filament Formation Enables Highly Cooperative Regulation of Human CTP Synthase 2. Eric?
Yeah, sorry. Yeah, thanks. So I'm a postdoc in the Coleman Lab at the University of Washington. And so I'm going to talk about how polymerization regulates the metabolic enzyme CTP synthase. Right, so filaments of CTP synthase are apparently universally conserved.
So we're not seeing your slides, sorry.
Oh, sorry. Seems critical. Oh, there we go. Right, okay. There we go. Okay, step one, right.
Excellent.
These filaments of CTP synthase are universally conserved. So they've been observed in species from bacteria all the way up to humans. And so basically what I aim to address was whether or not the structure and function of these different filaments are conserved across species. So most of what I'm going to show is sort of in vitro work, some biochemistry and enzyme kinetics, and structures determined by cryo-EM. And so in the way of introduction, so CTP synthase catalyzes the conversion of UTP to CTP. And so this is showing a monomer CTPS here. It's comprised of two domains, so a glutaminase domain, and that domain releases ammonia from glutamine, and then that's transferred to this ammonia ligase domain down here, where the ammonia is added to UTP to make CTP. And so importantly, this means that CTPS is heavily regulated, so it's sensitive to levels of all 4 ribonucleotides. And this sort of makes sense in that nucleotide balance is it's strictly maintained with imbalance in the levels of nucleotides leading to mutagenesis and disease. Although under certain circumstances, the levels of the 4 nucleotides, the balance is sort of shifted. So for example, during proliferation, we know that levels of CTP are elevated. And then in humans, there's 2 isoforms of CTP synthase, CTPS1 and 2. So CTPS1 is specifically upregulated in proliferation, and we have some initial evidence to suggest that it resists CTP inhibition, where CTPS2 is sort of the housekeeping version of the enzyme. So these 2 enzymes potentially filling those sort of 2 requirements I just discussed for sort of balanced nucleotide levels, and then with CTPS1 under particular circumstances, increasing CTP levels. And so that monomer I was just showing you of CTP synthase, so that constitutively assembles into dimers, and then in the presence of nucleotides, so either substrates or products, assembles into tetramers. And what I found is that binding of substrates or products to these tetramers drives 2 main conformational changes. So you can see these in these movies here. So upon binding UTP substrate, sort of interface of the tetramer sort of extends. You can see the positions of the monomers shifting relative to each other. And we also get this rotation of the glutaminase domains. So what's happening here is glutaminase domains are sort of rotating inwards upon substrate binding to become closer to the ammonia ligase domains. And so that conformational cycle is seems to be conserved. So we see it in basically all the CTP synthases we look at. And so it is conserved between bacteria and humans. But then polymerization adds another layer to this regulation. And so what I saw was that with human CTBS1, filaments assemble in the presence of substrates in the active conformation. And these filaments have increased activity. And then you see sort of the opposite effect with E. coli CTB synthase. So E. coli CTBS assembles in the presence of products in the inhibited conformation, and filamentation in this case actually decreases activity. And importantly as well, so these two filaments actually have completely different architectures. So they're both comprised of stacked tetramers, but they assemble by different contacts. So the E. coli filament has these two different contact sites, one in the glutaminase domain and one in the linker. Whereas the human enzyme assembles by this completely different helix in the glutaminase domain. And this helix is actually specific to eukaryotes, so it's not present in bacteria. It also contains this conserved residue H335, which when you mutate that, so changing just this one residue to an alanine completely abolishes polymerization. So these are just negative stain micrographs comparing the wild type protein to the non-polymerizing mutant. And as I mentioned, the CTBS1 filaments have increased activity relative to the free tetramers. And so preventing polymerization with this mutation leads to a big decrease in activity. And so then next what I did is looked at the other isoform of the enzyme, the human enzyme, so CTBS2. And we were really surprised to see that it seems to behave differently than CTBS1. So you see filaments both in the presence of substrates and products. And so we determined the structures of these as well. So the substrate-bound filament and the product-bound filament by cryo-EM. And both of the filament forms assemble by the same contact site. And this is the same contact site you see in CTBS1. So involving this helical insert with the H325 residue. But the tetramers within these filaments also undergo the same conformational changes I was describing earlier. And importantly, what this means is when you transition from the product-bound to the substrate-bound state, because you get this inward rotation of the glutaminase domains, it actually alters the position of the helical contacts within the filament. So this is sort of a view down the helical axis. You can see the helical contacts are actually rotating inwards. And what this means is the two filament forms have, again, similar architecture in that they're stacked tetramers. They have the same rise per subunit, but the rotation is actually different because of this rotation within the glutaminase domains. And so this made us consider that potentially, because the filaments are in different conformations, but they're assembling by the same contact site, that maybe intact filaments could morph between these two conformations without disassembling. And so to test this, what I did was to lock the filaments into, covalently lock them into filaments by engineering disulfide crosslinks at the filament interface. And so that worked quite nicely. So you can see down here in the presence of reducing agent, you don't have filaments. But then if you remove reducing agent, you get spontaneous polymerization, either with no nucleotides present or in the presence of substrates or products. And then we wanted to address, well, can these cross-linked, covalently cross-linked filaments switch conformations? And we're kind of fortunate in that even in relatively low resolution negative stain 2D averages, we can distinguish between the substrate and product-bound state on the basis of these differences in this sort of 180-degree repeat we see. So in substrates, it's every 300 angstroms, whereas in products, it's every 400 angstroms. And so indeed, if you look at the cross-linked filaments in the presence of substrates, we see this characteristic 300 angstrom repeat. And then upon addition of the product CTP, the cross-linked filaments can switch to the product-bound conformation with this different characteristic repeat distance. And all that to say basically that we think that this is what can happen. So this is just showing a movie of a CTBS2 filament morphing between the substrate and product-bound conformations. With the contact sites shown in orange here, as you can see that those sort of track within the filament. So you can preserve the contact site while switching between the two filament forms. Yeah, and then so want to then address, well, what does this do biochemically? So having these two filament forms, what are the consequences of that on the activity or the regulation of the enzyme? And so we compared 3 different conditions in order to do this. So looking at the wild type protein where you have filaments, the wild type protein where it's diluted below the critical concentration. So it's primarily tetrameric and you no longer have filaments. And then also the non-polymerizing mutant where again, you'd have tetramers. And what I saw is that unlike CTBS1, with CTBS2, the polymers didn't seem to have an effect on the maximum velocity of the enzyme. The Km for UTP was very similar. But then where we saw the major difference was when I looked at these CTP inhibition curves, where the wild type protein in filaments showed much greater cooperativity than the tetrameric form. So you can see that both in sort of looking at the steepness of these curves and also reflected in these Hill coefficients. So even for the non-polymerizing tetrameric form, the Hill coefficient approaches 4, with 4 being the sort of maximum, the theoretical limit of cooperativity for a tetramer. But then when you have filaments, you're able to exceed that, and so you get much more cooperative regulation. And I see the same effect. So upon diluting the wild type protein below the critical concentration where you no longer have filaments, again, you see a Hill coefficient around 4, so less cooperative. And then in the way of a model, so basically within tetramers, we get sort of classic cooperative regulation. So you can imagine you have a substrate bound active tetramer, and then upon binding CTP to one of these monomers, the conformational state of that monomer can influence up to sort of 3 of its neighbors. So it can induce cooperative changes within a tetramer. But then when you have a filament, in addition to these cooperative conformational changes within tetramers, you can also get them between tetramers. So you've essentially expanded sort of the cooperativity in these unbounded, sort of theoretically infinite polymers. And so this leads to enhanced cooperativity in the filaments relative to the tetramers. And so importantly, cooperativity would allow for sort of ultrasensitive regulation, which is important because it would allow us to stabilize nucleotide levels over a narrow concentration range. So basically providing tighter control over the balance of nucleotides. I just wanted to add quickly that, so these are the first sort of high resolution structures of the human enzyme. So they're allowing us to see sort of new details of the regulation. So just as one example, this is showing the CTP bound CTPS2 structure, which I now have at about 2.7 angstroms. And so looking at this, we saw that there's a CTP bound in sort of the canonical regulatory site here, but also surprisingly, we were able to see a second copy of CTP bound in an additional site, which hadn't been observed before, so that was very surprising. So the 2 copies of CTP, whereas previously we'd assume there were just 1 per monomer. And as I mentioned previously, the CTPS1 isoform appears to have much higher IC50 for CTP than CTPS2, and there are some sequence differences in this binding pocket. So we were thinking that perhaps this site may be lower affinity or completely absent in CTPS1, and that could explain the difference in those IC50s. And again, that could relate to their sort of physiological roles, with CTPS2 being sort of the housekeeping isoform that has sort of tighter regulation, and then CTPS1 being involved in proliferation and resisting feedback inhibition, allowing to expand CTP pools. Yeah, and so I'll leave it at that. And so just thank the lab. So Justin, my PI, and Joel and Quentin, who run our cryo-EM facility and are of incredible help, and just some collaborators here. So any questions?
Thank you very much. Really fascinating work, beautiful structural work. So as CTPS, it's not always in a filament, right? But when it's in the filament, it can switch between active and inactive. Is that correct?
When it's in a filament, yeah, exactly. So the filaments can switch conformations while remaining assembled, yeah, for CTPS2. We've at least
so like, what conditions induce polymerization? And then once it's polymerized, it's more sensitive, I guess more cooperative in being able to switch back and forth between active and inactive. But what conditions does it need this filamentation and this cooperative regulation?
You mean in sort of an intracellular context, or?
Yeah, exactly.
Yeah, I mean, that's not totally clear. So we know some of them that you see, like in yeast, for example, in starvation and things like that, you see filaments appearing. In human cells, I believe CTPS1 is known to polymerize during proliferation, and that's seen with some other filamentous enzymes. But that could be reflected again in their 2 sort of differential roles of the isoforms. So we would expect maybe with CTPS1, when you need to upregulate CTP production, you would see polymers. But this would suggest that if the filaments are necessary for sort of that tighter regulation. You'd expect to see CTPS2 filaments just during normal cell growth. But I don't know, I'm not sure if we've directly observed that.
Interesting. So let's see, we have a question here. Wei Chang Lin says, Dr. Lynch, your studies really inspire me. That's nice. I'm wondering if you can see bundle structure in fluorescence microscopy in vitro, a bundle structure.
Yeah, so I think probably what we're seeing in vivo when people look at fluorescence micrographs is probably bundles, given sort of the size of those structures. And we do see some bundling of the human proteins in cryo-EM. I don't have a structure of those, but they look ordered.
Even so, they come together, because there's some idea initially that these filaments could assemble really tightly packed, and that would make sense if they're all inhibited. But in this case, that's not strictly true. There's an active form as well. So do you see a difference in how the filaments come together, whether they're the active filament or the inactive?
Yeah, so it seems to be able to bundle under sort of both conditions, but we haven't really sorted out if there's any difference between, you can imagine differences in the regulation between single filaments versus bundles could be a possibility. So for example, sort of morphing between the two conformations, maybe that would be more difficult if you had bundles, for example. So I agree that that could maybe sort of lock things in a given state. Still looking at that. Yeah,
that's beautiful work on the cooperativity. So we have another question, Tom Huxford. Dr. Lynch, is there any evidence that metal ions, maybe zinc or even calcium, might be involved in stabilizing CTPS polymerization?
Ooh, yeah, I don't have any evidence of that. You have magnesium present just as sort of, because you have nucleotides around, but I haven't looked at any other metal ions, no.
Okay, great. I think there's another question in the Q&A, and watch in case new ones come up there, but thank you again.
Oh, sure. Thanks.
Now we'll move on to Jeffrey Peterson from Fox Chase Cancer Center with a talk entitled Enzyme Polymerization and Nucleotide Biosynthesis. Go ahead, Jeffrey.
I hope you're able to see that.
Yeah, that looks great.
Great, I'm gonna just put up a pointer. So thank you, Nancy, for the invitation to be here and for organizing this session twice, I guess. My lab works on two different polymerizing enzymes, both of which are involved in nucleotide biosynthesis. And what I thought I would do today is describe recent efforts and to try to assess whether enzyme assembly provides a fitness advantage in the context of metazoan tissues where they occur normally. And the first story that I'm going to tell you concerns the enzyme CTP synthase, which we just heard about from Eric's talk. And this image shows developing Drosophila eggs expressing GFP fused to CTP synthase. And while Eric's talk mentioned or focused really on the stacks of tetramers that can be assembled and studied in vitro, if we look in vivo, you can see structures as long as tens of microns can assemble in the native context, and exactly how assembly of these much larger assemblages in vivo impact the biological function of CTP synthase is still unknown. So we've been working on the Drosophila as an experimental context, and the ovary consists of a fundamental unit called the ovariole, and developing egg chambers are born at the distal end and then mature through 14 developmental stages until they are deposited into the oviduct. Now, each of these egg chambers consists of two primary cell types, the germline cells seen here in the center, surrounded by a follicular epithelium outlined in red. And CTP synthase forms filaments in both of these cell types, but that assembly is regulated developmentally in the germline cells. So in early egg chambers, very few CTP synthase filaments are seen, and as development proceeds, both the number and size of these structures increase until about stage 10, at which point each of the germline cells, and there are 16 at this point, have on average one filament. And this period during which CTP synthase assembles correlates temporally with the developmental period in which the germ cell nuclei undergo multiple cycles of endoreplication and also dramatically upregulate the ribosomal RNA. So this represents a really increased demand for nucleotides. And this is consistent with the in vitro biochemistry for this enzyme that is suggested as Eric talked about, for CTP synthase 1, that assembly is associated with an increase in catalytic activity, perhaps to satisfy this increased nucleotide demand. Now at stage 10, you'll notice all of a sudden filaments begin to disassemble. And this is despite the fact that CTP synthase protein levels remain high. And so we were intrigued by this carefully choreographed assembly process and wonder if it was providing a biological function in the ovary context to support egg production. And so to test this idea, we took advantage of the histidine 355 to alanine mutation that Eric introduced to his talk. And as he noted, 355 is located at the polymerization interface for tetramers, and mutation of that single amino acid disrupts tetramer stacking. And as you'd expect, also blocks the increase in catalytic activity seen when the wild type enzyme polymerizes. And so we introduced the 355 to alanine mutation in the context of Drosophila CTP synthase, and then expressed it as a transgene within the Drosophila ovary. In the control case, where we drive a transgene expressing wild type CTP synthase, we see that it forms morphologically normal filaments at the appropriate times during oogenesis. By contrast, when we express the H355A mutant, this enzyme does not assemble and remains diffuse. And in order to control for artifacts that could be due to integration site of the transgene, we validated this study with 2 independent transgenes for both the wild type and mutant transgenes. And the results are quantified here for 2 different stages in oogenesis, showing the profound loss of assembly of these large structures when they contain the histidine to alanine mutation. Now, this is, it's important to keep in mind here that these egg chambers are also expressing the endogenous wild type CTP synthase. And so we wondered whether expressing the H355A mutant had any impact on the assembly of the endogenous protein. And so to test this, we took advantage of a fly line in which GFP has been inserted into the endogenous CTP synthase locus, and then expressed the transgenes in that context. So again, in the control case, if we express the wild type transgene, we still see that the endogenous wild type CTP synthase polymerizes normally. By contrast, the H355A transgene completely abolishes assembly by the endogenous wild type CTP synthase. And again, that's quantified over here for both independent transgenes, showing the pronounced dominant negative effect of the mutant protein to suppress the assembly of the endogenous protein. And that makes sense if we can imagine that wild type and mutant co-assemble within tetramers, and consequently blocking the intertetramer associations that are mediated by that H355 mutant, blocking the assembly also of the endogenous protein. So now we're really poised to ask whether disrupting assembly has a phenotypic effect on egg production. So we isolated independent transgenes that gave similar levels of expression of either the wild type or H355A CTP synthase, either at the RNA or protein levels, and then took female flies expressing each of these transgenes within the germline cells of the ovary and mated them to wild type males, and simply counted the number of eggs produced over the course of 24 hours. And what we saw is that the H355 mutant that do not assemble showed substantially lower egg production than the matched controls expressing the wild type transgene, demonstrating a fecundity advantage given the ability to assemble CTP synthase into filaments. Now, while this experiment doesn't assess or establish what that advantage is, it does establish that there's an advantage to having that ability to form filaments. And we're working now on establishing this model, which is that assembly of CTP synthase during these developmental stages leads to increased egg production in order to support the production of eggs, and that disrupting assembly lowers CTP production and reduces fecundity. So now I'm going to move to the second enzyme that I wanted to talk about today, and that's inosine monophosphate dehydrogenase, or IMPDH. And this is an enzyme that sits at a critical branch point in purine nucleotide biosynthesis. IMP is a common precursor that can be used both for adenine and guanine nucleotide production, and IMPDH diverts it toward the synthesis of guanine-containing nucleotides. The enzyme forms octamers, which form nonpolar linear polymers in this way, in vitro, and which form larger assemblies in cells, analogous to what we've seen for CTP synthase. So a few years ago, we identified mutations that disrupt the ability of IMPDH to polymerize, and that led to our focus on tyrosine 12. And an alanine substitution mutation of that single amino acid disrupts IMPDH's ability to polymerize, both as a purified protein in vitro, as well as when expressed transiently in cultured cell contexts. So once we were able to disrupt assembly, we were of course asking, well, what consequence does that have for the catalytic activity? And we're rather surprised and admittedly disappointed at that time to find that IMPDH catalytic activity was just fine in the absence of polymerization. Now, recently, a biochemical function for assembly has been provided by work from the Bowie and Coleman labs, who independently showed an impact of assembly on a different aspect of the enzymology of IMPDH, feedback inhibition. So IMPDH is subject to classical negative feedback inhibition, where its downstream guanine nucleotide products can bind back and suppress the catalytic of IMPDH activity of IMPDH. And so in the filamentous form, IMPDH becomes more resistant to this feedback inhibition. And so as a consequence, in cells in which IMPDH is assembled, you can see, or you can imagine that the enzyme retains activity and generates higher levels of GTP before its activity is suppressed. And this would presumably allow for the accumulation of higher steady state amounts of guanine nucleotides compared to the unpolymerized form. So where does this actually happen in biology? And we identified the first metazoan context in which IMPDH occurs naturally, and that is in the context of T cell activation. So this is a study where we've taken primary murine T cells isolated from the spleens of mice and stimulated the T cell receptor ex vivo, revealing this stimulant-dependent assembly of IMPDH into filaments. And this is accompanied by an increase in the steady state pools of guanine nucleotides in this context. So now we had both a way of disrupting assembly and a context in which it naturally happens. And so we wanted to know whether assembly is important for the functional consequences within T cells. And this is ongoing work, but the approach that we took was a little bit different than where for CTP synthase, we used expression of a dominant negative form of the enzyme to suppress assembly of the endogenous. For the IMPDH, we instead collaborated with Naira Akizu at CHOP in Philadelphia to genomically edit, using CRISPR technology, the endogenous murine locus for IMPDH2 to generate the Y12A allele in the mouse genome. And Naira's group was successful in doing this and then bred these mice to homozygosity. The mice are viable, and when we isolate T cells from those mice and stimulate them ex vivo, we see, as expected, that assembly is disrupted, both in the homozygous case as well as in the heterozygotes. And this validates this, the presence of the mutation and the fact that we've now disrupted IMPDH assembly. Now, the mice that are homozygous for Y12A are born in normal Mendelian ratios and thus far have lived a happy 1-year life, telling us that assembly of IMPDH2 is apparently not essential for life. And so we're now characterizing these mice in more detail to try to understand whether they differ, and thereby whether there's a function associated with assembly. And so one of the things that we did was to use flow cytometry to characterize the adaptive immune system of these mice. And we find that CD4, CD8, and gamma delta T cells are present in normal ratios in the mutant and wild type mice, telling us that at least development of the immune system also doesn't depend on IMPDH2 assembly. But what we're really interested in is the functional consequence for adaptive immunity, because existing immunotherapy agents, mycophenolic acid, are potent immunosuppressives that act by suppressing IMPDH catalytic activity. So in ongoing work that I'll have to tell you about next year, we are challenging these mice to viral infections to assess the quality of their responses in the wild type and mutant mice. So my time is up, so I'm going to move on to sum up with one thought, and that is biological context is everything. We're trying to move beyond the in vitro biochemistry and cell-based studies to explore whether assembly is providing a functional context in the native tissues in which assembly occurs. So for CTP synthase, we focused on the ovary where a filament assembly occurs naturally, and used a dominant negative transgene to suppress assembly to reveal a role for CTP synthase assembly in egg production. For IMPDH, we've taken a different approach to genomically edit the endogenous locus to generate only non-polymerizing IMPDH2. And that's so far revealing subtle phenotypes that suggest that IMPDH assembly may be important only in specific contexts and tissues. So with that, I will acknowledge the members of my lab who contributed to this work, my collaborators at Fox Chase, and the Coleman and Akizu groups for the work that I tried to highlight along the way. And thank you very much for listening, and I'm happy to take any questions.
That's great, really beautiful work. No one can question how important this is in vivo. It'll be exciting to see the results of the viral challenge to the mice next year.
We're hoping to get back in the lab soon.
Yeah, I know. Yeah, we're back here in Arizona. It's a funny state. Okay, so we have some questions. So Hanil Yu for Jeffrey says, beautiful images. Do the eggs with defective CTPS hatch, develop into viable larvae or flies and flies?
They do. We do produce progeny from those eggs that are laid.
Right. From Nagi Ayad, does CTP synthase levels increase in mammalian development? And is it localized to reproductive structures?
That's a great question. We've obviously focused on the Drosophila ovary because of the genetic tools we can use in that context, but we have not looked at mammalian development.
That's great. Let's see. Another question from Wei Chang Lin. Dr. Peterson, thank you for your nice talk. H355S mutant represents active or inactive enzyme activity in the egg chamber.
I assume you mean the H355 alanine substitution that we generated. That enzyme is active, but it doesn't show the increase in catalytic activity on polymerization. So there is basal activity, but you don't see the enhancement on assembly.
Can it be measured in the egg chamber?
In the activity, it can, in principle. It's something we've tried to work on doing isotope tracer of precursors, but it's technically challenging to keep this tissue alive ex vivo and do those sort of experiments, but that would be a great experiment to do.
We have a comment here from Gaurav Kumar. Dr. Jeffrey Peterson, I wish you the best of luck for your future work. It's true that biological concepts are everything. Wonderful work. Wow, that's nice.
Thank you so much. Appreciate that.
Okay, so let's see, I think there are a few unanswered questions in the Q&A box that other speakers haven't answered yet. Okay, we have another question. Shriram Venkatesan, a few other talks in this session alluded to how enzyme activity may be better when present in the filamentous form. Would you think your mechanism of suppression of negative feedback be a general theme in such cases?
Please.
Yeah, so forming the filament to kind of ignore feedback.
I think that that's, I think it's highly likely that's also going to be a phenomenon observed for other enzymes. It seems unlikely to be unique to the IMPDH case. I think it really is interesting to note how distinct enzymes utilize assembly to modulate enzyme activity in such different ways.
Yeah, it's usually the more regulated the enzyme, the more likely it filaments, it seems.
Yes.
The most highly regulated, another layer of regulation this way. Okay, well, I'd like to thank all the speakers today. Excellent talks, and thank you again for participating. And thanks to the participants who came and were able to be involved and watch the talks and ask questions. So I think now I need to turn it over to the ASBMB host.